afternoon. My name is Marlie McClung. I'm a sophomore here at Northeast Mississippi Community College, and today I'll be presenting on Drosophila Melanogaster, more commonly known to us as fruit flies. So today I am going to be presenting a presentation that I formulated this fall with my professor. My research expounded upon the former students that he had that were members of our Honors Institute. They found that different mutations had limiting reproductive abilities, and specifically that the white eyes, denoted by the W on the screen, limit the reproduction. So with that being said, my research was to either prove or disprove that the white-eyed flies were limiting in reproductive capability. So first, let's discuss a few factors that could influence my results and what I see. We'll first focus on the white males, denoted by the W right here. White males with the white eyes are potentially going to be dazzled by natural light. This is because the lack of pigmentation in their eyes causes them to be more prone to temporary blindness whenever the natural light is coming in on them. Because they are blind, it often leads to male-to-male courtships because they mislocalize this white protein that is found on the females. Let's talk a bit about the process. This image here on the left is from the University of Washington, where they did a Drosophila Melanogaster study on a much larger scale. The females lay up to 100 eggs per day. They have a 12-day period from the time the eggs are laid to the actual emergence. This chart on the left gives a visual representation. So when I would perform my research, I had to go into the lab in the mornings, take the vials of living flies out into the environment and release them. Once I did that, I returned the stopper to the vial, returned the vials to the lab, and I returned to the lab 8 hours later. Then any flies that I found within the vials I knew would be virgin flies. I examined the virgin flies to determine whether they were male or female, and then placed them in a 1-to-1 male-to-female ratio in vials with media and yeast so that the flies could thrive. Let's talk a bit more about my methodology. On the right here, I have another picture from the University of Washington with an example of a Drosophila vial. Anytime that I reference the wild type, I'll be using a plus sign, and anytime I reference the white-eyed, I'll be using a W. I would also like to denote the 4 crosses that I had to repeatedly perform in order to obtain substantial results. I performed a cross of the wild white-eyed female by the wild-type male, the white-eyed female by the white-eyed male, the wild-type female by the wild-type male, and lastly, the wild-type female by the white-eyed male. Now, it's essential that we discuss our outcomes. Within any project, you're going to see some failure, and it's necessary that you acknowledge that and pose the question as to why it failed. So first I'll talk and discuss the successes. I had 29 successful crosses yield from the ones that I performed. However, I also had 40 failures. You could answer this question of why it failed in many ways. For example, my professor and I examined some of the failure vials and found that it was a female-to-female cross or a male-to-male cross. Crosses like that are not going to yield reproduction. Here we have the results. So in the wild-type female by wild-type male, I had 6 successful crosses. Each number in the column represents the number of flies that were in that particular cross. So for example, I had a cross with 147 flies. I had a cross with 84 flies, and so on. This column has wild-type female by white-eyed male. I had 6 successful crosses from that batch. This one is white-eyed female by wild-type male. Here I had 8 successful crosses. And lastly, white-eyed female by white-eyed male. Here I had 9 successful crosses. Now this is an ANOVA. ANOVA is an operation performed within Excel that yields an analysis of variance. Some main statistical measures I'd like to denote from this slide are the count. This is the number of successful crosses from each batch. The sum. This is the total number of flies that came from each batch. The average. That was the average number of flies among all the crosses in that batch. And then lastly, the most important on this slide is the p-value. I'll get to what the p-value stands for and represents a few slides away. Here again, I would like to just mainly point out this p-value of 0.0149. So in any experiment, the p-value is the percent chance that this happens by random. The alpha p-value is 0.05 standard. This means that anytime there is a 0.04 or less, that means that more than likely this didn't just happen by random chance and that there's an explanation for this difference. So the rejection of the null hypothesis suggests that the average is not the same for both of the groups. Here it breaks down when you analyze the wild-type female by the white-eyed male and the wild-type female by the wild-type male, it accepts the null hypothesis. Let's go to the next slide and look at the actual figures to determine just exactly how that works. So 0.229 is obviously much larger than 0.05. Therefore, it accepted. But here you have 0.009, which is much smaller than 0.05. Therefore, that was a rejection of the null hypothesis. Moving forward, unfortunately, the wild-type male was identified as a common denominator limiting reproduction. While this did contradict what former researchers suggested, there are many plausible reasons for why this occurred. There could be a unique deficiency limited to my specific subgroup of flies. Or the fact that my professor and I knew that temporary blindness could affect the flies and move the flies to the back side of the room where there was not as much natural light could have caused an influx in the number of white-eyed. Therefore, we wouldn't have seen limits. So, although my results were very unprecedented, there can be an explanation that can be determined. From my research, we rejected the hypothesis that the white-eyed flies were limiters. However, my professor's future honors students can easily expound upon my research and analyze the flies that I dealt with. Hopefully, they will look at this particular group and determine whether they do have some kind of unique deficiency that can be handled. Thank you.